make some time, even if you are working, you have to make some time to go to the laboratory. We have a 14-day session, okay, that is for 2 weeks. You have to afford some time to go to the laboratory wherever your study center is and do the experiments. Of course, there are some of the protocols which need to be followed for this laboratory course, which I am coming. But this is, at the preliminary level, I would like to mention this, that it is very important and you have to attend the laboratory classes. Now, coming to the next point, that is laboratory course, as I said, is the first, lab course 1 is the first laboratory course in MSc Zoology program based on the 4 theory courses that you have studied in semester 1. So, in total MSc program, you will have total 4 laboratory courses because you have 4 semesters. And you will get an exposure of several experiments from different courses that you will be studying in your theory, as I already told you. So from each theory, there will be certain number of experiments so that we can cover almost all the theory papers in practicals. These experiments are intended to provide hands-on training and develop your skills of observation, analysis, and interpretation that will help you in your dissertation. This is another very, very important point, that in semester 4, you will have to undertake one project, and there is one paper, dissertation, which will be mostly based on some of the experimental works that you do, and you have to submit a report, that is project report. You have to attend the laboratory wherever you are working, and based on the work that you have done for a certain time period, which will be informed you will be required to submit a report. And maybe you might have to deliver one presentation, or you might have to face a viva voce examination. So that is very important. So until and unless you do the lab courses very carefully, you will not be able to perform good in the dissertation. So it will help it in your dissertation. And as I told you, there would be many of you who would aspire to be a researcher, to do your PhD, and to do maybe in future after your PhD, some of you might even go for your postdoctorate either in India or outside India, and maybe you would like to see yourself in future as a scientist in reputed institutes. Okay, the different national institutes that we have all over India. So you may like to see yourself as a scientist. So for all these, this laboratory course will be very, very vital to shape your career more properly. Laboratory course 1 is an intensive course which you will need to complete over a period of 1 week. This 2-credit laboratory course requires 60 hours of input by the learners. You will have to work 8 hours each day with 2 laboratory sessions of 4 hours each, where 3 hours will be allotted video viewing or demonstration by the counselors, and 1 hour for preparing your record notebook and viva voce undertaken by the counselors. So there will be a total of around 14 sessions. Now, a schedule for laboratory experiments will be given to you by your counselor in the first session. Make sure that you do not miss any of the laboratory session, since it will be very, very difficult to repeat the same experiment due to time constraint. I already told you that 24 experiments are there, and each experiments are from different theory courses. So each of them are important, and therefore time is short. So it may not be possible for the study centers to repeat the same experiment for any particular student, or even 2 or 3 students. So please attend the laboratory course and try to make best use of it. You ask questions. Okay, try to have an inquisitive mind And even if you don't get chance of doing it in your own hands, at least observe the experiments very carefully and try to ask questions and make best use of these practical sessions. So the course comprises one block containing in total 24 experiments, and this course, laboratory course 1, is already available in our e-Gyan-Kosh. You can go to the e-Gyan-Kosh and you can download the entire block for this laboratory course, and I, being the course coordinator of this course, I have completed it long back and it's available in e-Gyan-Kosh. Now coming to the experiments, if I give you an overview now by picking up each of the experiments. Experiments 1 and 2 will be some sort of recapitulation. Why recapitulation? Because you will see that in experiment 1 and 2, because you are just facing the laboratory experiment at the MSc program for the first time. So maybe we want you to just refresh whatever you have learnt in your undergraduate program. For instance, most of you, or I think whoever is taking this program, we considered that you are coming from zoology background only, or maybe botany background. That is, you have done your BSc in zoology with botany, zoology, chemistry, maybe the combination. So you are already introduced to the preparation of chromosomes. That is cytological preparation. That is observation of mitotic chromosomes and meiotic chromosomes. Because these are not very difficult experiments, and even in the college level, in the undergraduate level, these are being taught. How to observe a mitotic chromosome, especially the metaphase chromosome. Because you all know that metaphase stage is the stage where the chromosome is most clearly visible. So how do you make a chromosome preparation from, let's say, root tip cells, like root tip or shoot tip? Or if it is a meiosis, then you can study the meiotic chromosome from pollen mother cells, or you can observe from the grasshopper testis, if it is an animal system. So if you see in the first experiment, we have actually tried to make you acquaint with the meiotic chromosomes, which you will be preparing from the grasshopper testis. And in experiment 2, you will be studying the polytene chromosomes. You know, the salivary gland chromosomes of Drosophila, where you can actually, that is actually a best tissue or the best system to observe the polytene chromosomes. So experiment 1 and 2 will be like some sort of recapitulation, which probably you have already done in your undergraduate, that is BSc program. Now experiments 3 to 7 will include some basic experiments of biochemistry, including buffer preparation, amino acid titration, characterization of enzyme kinetics, and spectrophotometric assay to estimate the level of certain biomolecules. Because if you see, you have a paper on physiology and biochemistry. So we have actually in this experiment 3 to 7, we have actually included experiments from biochemistry. Now whenever you are talking about biochemistry, the first thing that needs to be learned is how to prepare a buffer. I already told you. Like in biology, we normally use the tris buffer, or you use the phosphate buffer. So phosphate buffer, it is actually a combination of disodium hydrogen phosphate and sodium dihydrogen phosphate. But you have to know in what proportion you will be mixing the two to get, let's say, a phosphate buffer of 1 molar strength of a certain pH. If I ask you to prepare a buffer of pH 8, how will you prepare? Or let's say a tris buffer. Okay? How will you prepare a tris buffer, tris HCl, which is a very common buffer in biology? How will you prepare if I ask you to prepare a tris Cl buffer of pH 8 or pH 7.5? How you are going to prepare? So that kind of training you will have in this experiment, in these sets of experiment. Then enzyme. That is when there is an enzyme substrate interaction, and when the enzyme degrades its substrate, then what is the kinetics? Enzyme kinetics. So you must have studied, or you may be studying, the Michaelis-Menten equation. Okay? So how to calculate or how to characterize the enzyme kinetics? How will you study the enzyme kinetics? Taking any representative enzyme, how will you study the enzyme kinetics? And also, you know that enzymatic reaction, it is dependent on several factors. It depends on temperature, it depends on pH. So if you change these factors, if there is a change in temperature, if there is a change in pH, then how these kinetics actually change? How will you show it experimentally? So these will be actually, you will be exposed to this kind of training here. Biomolecules. What are the common biomolecules? Protein, sugar, lipids. Apart from that, there are certain vitamins, amino acids. So if somebody asks you, okay, you are given an animal cell, what is the amount of a particular amino acid? Let's say tryptophan, let's say proline. Or somebody asks you, what is the level of amino acid, or what is the level of protein which is present in that sample? So you should have some protocol, or you should have some methods by which you can estimate that. And that estimation is based on one instrument which is known as the spectrophotometer. And in most of the cases, this spectrophotometric measurement or spectrophotometric assay of any metabolite is based on one principle which is called as Beer-Lambert's law. So you need to learn all this, you need to learn the principle, and then you need to learn how will you estimate this using the spectrophotometer. How will you measure a protein level, let's say in any animal cell, or maybe even in a plant cell? How will you measure the protein level? How will you measure the amino acid level? Or let's say vitamin C level. Vitamin C is a very, you know that it's a very important antioxidant which we have in our body, and vitamin C is also supplied in fruits, green vegetables. But there are certain levels of ascorbic acid in our body as well. And what is that level? So these kind of experiments are there, which are very interesting. You will try to correlate it with our daily life. You can correlate it with daily life, because if there is a change in any of these metabolites, there can be a deficiency. You know what will happen if there is vitamin deficiency, or if there is protein deficiency. We all know the consequences. Therefore, a proper level needs to be maintained in the body. But how will you ensure that the proper level is there in the system? For that you need to measure. And therefore these practicals. Experiments 8 and 9 will focus on the preparation of blood smears and determination of total and differential leukocyte count. What happens when a doctor prescribes you that whenever there is any disease, the doctor prescribes you a blood test. So what do you measure? What do you actually see in a blood test? You see what are the different cells, what is the proportion of each of the cells, the red blood cells, the WBC. Among the WBC you have the granulocytes and agranulocytes. So the doctor actually tries to see that what is the percentage of each of the cell, whether it is normal percentage or whether there is any anomaly, whether there is any abnormality. Certain cells are increased more than the other. Like if you have anemia, there is lesser red blood cells. Hemoglobin becomes lower. All of us know that. But for that you need to have some experiments. That is based on the blood smear. And then you have to take the counts. I mean total population of the cells, and among that total population, percentage of each of the cells. So I think you can understand what is the importance of this experiment, because it is connected to our real life. Experiments 10 to 15, you should be very careful, because probably for the first time at your master's you will be introduced to molecular biology experiments. And molecular biology experiments and cell culture experiments need special attention, because you cannot use or you cannot use the chemicals, buffers and the different glasswares and plasticwares without sterilization. You have to sterilize each and every component, buffers, the containers, the conical flasks, beakers, whatever you are using, measuring cylinder, that has to be sterilized. And for sterilization you have one equipment which is called autoclave. So each of the components need to be autoclaved. There is a particular principle of autoclaving which you need to learn, and you need to also the same principle as a household pressure cooker. It works on absolutely the same principle. So there is a particular temperature, there is a particular time for which needs to be maintained for sterilization process in an autoclave. So that has to be there, and the laboratories which are normally giving training in molecular biology and cell culture need to have an autoclave. So here what you will learn in molecular biology experiments. So these are mostly based on theory courses which you will learn in molecular cell biology, paper 1, and genetics and animal biotechnology, that is paper 2. Now here you will learn certain techniques like how to isolate a plasmid DNA. That is, you grow the bacterial culture, and then from the bacterial culture, if the bacteria already has a plasmid, how will you isolate the plasmid DNA from the bacterial cell? Then after isolation, you need to check whether you have isolated the plasmid DNA properly. For that, there is one technique called agarose gel electrophoresis. So that is another experiment. When you need to clone, when you need to introduce any foreign gene, right, in a system, then you need to cut the vector DNA. And therefore, you need to cut the DNA with one enzyme which is popularly known as restriction enzyme. So another experiment will be where you will learn how to digest a vector DNA using a restriction endonuclease. Any restriction endonuclease. It can be EcoR1, it can be Hindi3, BAMH1, whatever is available. How to cut the DNA. The next step in gene cloning is to ligate, that is to undergo ligation, where the vector and the foreign gene, they are joined. And this joining process is known as ligation. Therefore, there is one experiment where you will be taught the ligation of the vector DNA and the insert DNA. Insert means the gene which you are going to insert into the vector. So how will you ligate it? After ligation, you need to introduce it in a bacterial cell. Which is the bacteria that you normally use in a laboratory? That is Escherichia coli. So you need to introduce the plasmid DNA in a bacterial cell. And what is that process called? That process is called transformation. And therefore, another experiment is there where you will learn how to undergo this transformation experiment. For that, you have to prepare the transformation competent bacterial cells. So how to prepare bacterial cells competent enough to uptake the DNA, and then how do you add the DNA, and finally the DNA is being incorporated into the bacterial system. So this will be another experiment. So you see that you get a total combination or total package of experiments from 10 to 15, where you will cover almost all the steps which are followed in a gene cloning process. That is, cloning of any foreign DNA into the vector system. And the details of it will be covered in your paper number 2, that is Genetics and Animal Biotechnology. You will be studying the theory part in this paper or in this particular course. Experiments 16 to 20 will deal with experiments on cell biology, where you will get an idea about the aseptic techniques and steps of animal cell culture, including the different forms of culture media, in addition to cell viability and DNA damage analysis. Cell culture, that is, you have to use a particular media, an artificial media, where you will culture the cells, that is, grow the cells. So the culture can be in a media, it can be liquid media or it can be a solid media also. So you culture the cells. So one experiment will be on cell culture. Another experiment will be where you will be taught the different aseptic techniques. Aseptic technique means, that is, whenever you are carrying out a tissue culture or cell culture experiment, then you need to have a very protected kind of environment so that there are no contamination. There should be a separate cell culture room where you will be able to carry out these experiments. It should be free from dust, contamination. Okay? So, aseptic technique. This is known as the aseptic technique. Next, supposing whenever a cell grow, there will be cells which will undergo cell damage, or a cell damage may be due to different physical factors, different genotoxic stress, ultraviolet radiation, or there is any kind of, let's say, chemical which actually induces DNA damage in your system. How will you measure that? That in your system or in any animal system, the DNA has undergone damage. Okay? So for that we have one assay protocol which is called COMET assay. So there will be an experiment on COMET assay. And then you will measure the cell viability. There are experiments where you will measure the cell viability or cell death. Like, you know that apoptosis. This is one of the topic that you will study in your molecular cell biology. I think it should be in unit 16, that is cell death and renewal. So how will you understand, or how will you demonstrate whether any cell is a normal cell or an apoptotic cell? That is, the cell is undergoing cell death. So you should have some protocol, or you should have some method to understand whether it's a normal cell or it is a dead cell, or it is progressing towards cell death. We have experiments on that. Finally, the last 4 experiments, as I already told you, they will be related to parasitology, because the 4th paper that you have in theory is the parasitology. So here you will be mostly observing specimens of arthropod vectors, learn about the life cycle of different parasites, Plasmodium, Trypanosoma, identify the different species of Plasmodium and study the different stages of life cycle of mosquito as a vector, because mosquito as a vector, it causes so many diseases, malaria, dengue, right? These studies will be mostly based on photographs, charts, microscopic slides and museum specimens. So you will be shown maybe slides, permanent slides, or you might be they might show you some museum specimens, or they might take you to some place where you have the collection of such specimens which you can observe and you can record your observations. So these are the purposes, these are mainly the total overview of what you are going to do, what we actually expect you to learn based on the syllabus of the laboratory course 1. Now coming to the assessment part. So long you were learner, but now you become now you will be examined what you have learned. So take it very seriously. So you will be assessed for your performance in the laboratory course during the entire period. It is a continuous assessment. So each day what you are doing, that will be observed by whoever the counselor is. So make sure that you are attentive. Your counselor is supposed to give you proper instructions and necessary guidance to carry out the experiment, as well as assessing your performance simultaneously. Therefore, pay proper attention to the instruction and maintain proper laboratory ethics. What do you mean by laboratory ethics? Laboratory ethics means even when in future if you go to any lab to work, there are certain things which you need to follow. What are those? Number 1, you should not take your food sitting in the laboratory. Why? On one hand, you are actually contaminating, or you are actually you are not maintaining the proper protocol or proper cleanliness issue. Second point, in a laboratory, there can be certain chemicals which may not be very pleasant for your health. So if you are taking your food by sitting inside your laboratory, chances are that your food might get contaminated and you consume something which is contaminated, which is not safe for your health. So please develop a habit of taking your tiffin or taking your food or whatever, by not sitting in the laboratory. Another point, very important, handle all the instruments very carefully. Microscopes, any other instrument, It can be a centrifuge machine, the spectrophotometer, the micropipettes which you will be using in molecular biology work. Because most of these instruments are very costly. Okay? They cost in lakhs. And once an instrument gets damaged, it is very difficult to, undergo the repair process. So what will happen is the experiments will suffer. So please, whoever the instructor or the counselor is, whoever is showing you, please follow him or her. And until and unless you are sure about the operation of the machine, please don't handle it. First learn it. Take your time to learn. And then handle the instruments, if at all you get the opportunity to handle. Otherwise, please pay attention to the demonstrations that are going, so that in future you learn the principles of each of the machine, operation of the machine, so that in future you can handle it. So this needs to be very carefully followed. Now, upon completion of the course, you will face a term-end examination on the final day based on the assigned experiments that you have performed during the entire tenure. 70% assessment will be made on the guided experiments, and the rest 30% will be on the assigned experiments. So maybe you will be asked few questions also. Maybe they can ask you some viva voce questions which you have learned for the time period based on the experiments that you have been taught, the basic principles, the operations, sometimes the calculations. Okay, and in that way you will be assessed for your laboratory course. So it is a continuous assessment, and also on the final day you will have to face an term-end examination. Now, next part is the study guide. So as I told you that before you enter your laboratory for performing experiments, you should go through the experiment thoroughly as given in the manual, because as I told you, it is already available in e-Gyankosh. So you can download it, and before you go to the laboratory each day, you must read the instructions which is given in your manual. You should specifically spend more time on the procedure and the observation part, that is the methodology or the procedure part. What are the steps and what you are expected to observe. So these two things you have to keep in your mind before you are going to the practical classes. You should underline the important steps given in the laboratory manual. Please handle all the laboratory instruments very carefully, as most of them are quite expensive and need adequate fund for repair if damaged. So it becomes very difficult once an instrument gets it shuts down. For certain experiments, you need to carry dissection kit containing scissors, needles, scalpel, forceps, slides and cover slips, like experiment 1 and 2. Think of experiment 1 and 2, where you need to prepare the chromosomes, the meiotic chromosome or the polytene chromosomes. So you need to carry biology box, right, with all these watch glass, the slides, cover slips. For experiments in molecular biology and biotechnology, it is advisable to wear apron and use gloves. I think for these experiments it's better that you wear apron, because as I told you, cleanliness is a must. Similarly, for cell culture experiments, you need to keep the working space clean and maintain sterile conditions. Like in many study centers or in many educational institutes, they have a separate cell culture room where there is a laminar hood. Or even in plant tissue culture, you have a laminar hood. Okay, so there you should maintain utmost cleanliness. You have to wipe the laminar hood with, let's say, alcohol before you start your work. There should be a spirit lamp. The flame should be there, okay, where you actually flame sterilize the conical flasks or whatever, beaker or mostly the conical flasks. You sterilize them. Like if you need to inoculate something, even the inoculation needle, that has to be sterilized. So what I mean to say is it should be a very sterile condition, and that has to be maintained. Be cautious with the use of reagents and chemicals, especially which are corrosive, toxic and inflammable. You should be very careful. Like ammonia solution. If you are working with, let's say, formaldehyde, or let's say if you are working with ether, you have to be very careful. Even for hydrochloric acid, entire room might get, if you leave it just like open, entire room might get filled up with fumes, even for ammonia. So please take care of this thing, and especially which are inflammable, which catches fire. Be very careful. Alcohol, even alcohol, the absolute ethanol or methanol. Be careful. Do not forget to carry laboratory manual and a practical record book for recording your observations. Please make extensive and detailed notes on the observation and results of the experiments performed. Please note the day-to-day observation in a notebook, because that will help you in future. What you have done in your laboratory session, what you have learned, those protocols, because that will help you in your dissertation. Interact with your counselors to the maximum during your stay at the study center. Mind that it is always wise to ask a foolish question rather than sitting idly. If you think you are clever and you know everything and you keep quiet, you are not going to learn. If you ask foolish questions, you will be able to learn. That means you are wise. Remember this. So please ask questions. Cultivate a habit of asking questions, however simple the questions are, because then only we learn. Develop a habit of asking questions, however simple they might be, and bring out the best of your counselors. Try to learn as much as you can so as to make the laboratory course meaningful. Don't make it a center for gossip and chit-chat. Make it meaningful, make the sessions meaningful, learn something, ask questions, and then only you will enjoy your class. So surely the hands-on training will help you to also understand the theory courses better. Because in theory, okay, fine, you see the diagrams, okay, and you write, but if you see those in reality, you see the bacterial cells growing, you see the animal cells growing, you see the chromosomes in reality, then it is more exciting. And that is why science is so exciting. Because, especially the life sciences, because you get to see them in reality. So try to make best use of it. And it will make the theory courses more interesting, okay, more attractive for you. Now coming to the objectives. I think this is the last part that I am going to cover, that is the course objectives, before we will close today's sessions. As I already told you, since this is the first my first Gyan Darshan session with this program and also with this course. So in the future, maybe in the future sessions, I will take up the experiments one by one. But today, my main intention was to give you a total overview of the purpose of this laboratory course. So objectives: after undergoing the experiments of the laboratory course 1, you should be able to identify various stages of meiosis, describe the morphology and structure of the polytene chromosomes, prepare some of the common buffers used in analytical biochemistry, plot the experimental titration data and interpret the graphs to determine the approximate pKa values. I will be discussing on each of these experiments in the later sessions. So for the time being, just take a note of what you are going to learn. Describe the various properties of the enzymes, estimate sugar, amino acid, vitamins and nucleic acid, estimate the level of glucose and cholesterol in blood, and prepare a slide with human smear. Estimate the total leukocyte count and differential leukocyte count. Isolate, purify and visualize plasmid DNA. Visualize the DNA through agarose gel electrophoresis. Determine the quantity and purity of isolated DNA or RNA. That is mostly based on spectrophotometric determination. Set up a reaction for restriction enzyme digestion of plasmid DNA. Like you will have to use a buffer where you need to add the DNA and then the restriction enzyme, and then set up a reaction. The reaction is normally set at 37 degrees centigrade. Set up a ligation reaction maintaining all the necessary condition. Prepare competent cell and undergo transformation of recombinant plasmid in the competent bacterial cell. Even it may not be recombinant plasmid, it can be normal plasmid also, where there is no gene. You just want to introduce a plasmid in a bacterial cell. That is also possible. That is through transformation. Describe the various techniques or procedures followed for maintaining aseptic conditions in the laboratory. Carry out the steps of mammalian cell culture. Measure the cytotoxicity levels using MTT assay. Measure the cell viability by trypan blue staining or dye exclusion assay. Trace the extent of DNA damage using COMET assay. Explain the peculiar features of common and well-known arthropod vectors. Explain the various stages of life cycle of Trypanosoma gambians and Ascaris lumbricoides. Identify the various types of Plasmodium species infecting humans through change in morphology of red blood corpuscles in human blood samples. And discuss about the 3 important mosquito vectors, that is Aedes, Culex and Anopheles. So objectives are mostly based on the different experiments, as I have already discussed. So today, this was, I think, sufficient for today. I think I have been able to at least introduce you today to this laboratory course 1. And please immediately, if you have not downloaded eGyankosh material, that is laboratory course 1, please download it and you start. At least you read the manuals, read the protocols and observation part particularly, and try to find out what you need to learn actually, what you need to do, perform, and what you expect to observe after doing the experiments. Because if you have a knowledge beforehand, then finally when your classes starts, when your laboratory session starts, it will be very much easier for you. So I think that is all for today. And I wish you all a very, very happy learning, and I wish you, whoever have enrolled for this program, this is a new program, and we really look forward that our learners, they make best use of this program, and we would like to see that this program helps you in properly shaping your career in future, in terms of your job opportunities whether at the teaching level or at the research level or even at the industry level. Because when you are doing masters, we expect a lot from you, because this will help you in your future, in your career prospects. So I wish you all a very, very happy MSc program and a very, very happy learning these 2 years, coming 2 years. And we will keep on meeting in the subsequent Gyan Darshan sessions for the details of the laboratory experiments. Thank you all.